basically in this lecture are the amino acid. Now, amino acids, like I said, are the monomers of protein. So we can say that the amino acids are the basic structural units of proteins. Each amino acid contains an amino group and a carboxyl group in the molecule. So for instance, now the general structure of amino acid is you have a carbon atom, and then you have hydrogen, and then you have NH2, and then you have let me say here you have COOH, and then here you have the NH2, or let me put the NH2 here, and then here you have the hydrogen, and then you have arrow group. Now this arrow group here is what is called the side chain of the amino acid. The arrow group is called the what? The side chain. So this group here is what we call the side chain of the amino acid. All right, side chain of the amino acid. Why? This is the amine group, and then of course this is the carboxyl group. So for every amino acid you see, it contains this carboxyl group and then the amine group. Now the carbon atom of the amino acid to which the functional group are attached to is known as the alpha carbon. So this carbon atom here is what we call the alpha carbon. Every other carbon atom you can say alpha, then you have beta, gamma, and whatsoever. Now this side chain is what distinguishes one amino acid from the other. This side chain is what distinguishes one amino acid from the other. Now in solution, let's say in neutral solution, amino acids are mainly in the form of dipolar ions that are called Zwitter ions. So in solution, amino acids are in the form of dipolar ion called Zwitter ion. Now I will not tell you how these Zwitter ions are formed. At your level, just know this. When you get to 200 level biochemistry, you understand better. So how is the Zwitter ion like? Now the Zwitter ion is dipolar. Now when you say something is dipolar, it means you have the positive charge and then you have the negative charge. Now how is it like? If you protonate this group here, it's going to give us NH3. Here will be positive, and then of course you have carbon, you have hydrogen, and then here will be COO minus, and then you have arrow, which is the side chain. This arrow is the side chain, like I told you. The side chain is what distinguishes one amino acid from the other. This form of the amino acid is what we call the Zwitter ion form. It's what we call the what? The Zwitter ion form. So you should be able to write amino acid in their Zwitter ion form. Now generally, there are about 20 naturally occurring amino acids. Of course, there are others, but there are about 20 naturally occurring amino acids. Now, these 20 naturally occurring amino acids group themselves together to form the proteins that we have in living system. Are you getting it now? There are 20 naturally occurring amino acids. Now, it is expected that out of these 20 naturally occurring amino acids, about 10 of them, not up to 10, but let's just take it, 10 of them are essential amino acid. And then we have another 10 that is non-essential amino acid. Now, what are the differences between the essential amino acid and the non-essential amino acid? Now, the essential amino acids are those amino acids, right? That the essential amino acids are those amino acids that are present in our body. Are you getting it now? Present in the human body. Are you getting it now? And hence, we don't need to take them in as diet. Why the non-essential amino acids are those that are not present in our body, so we have to take them in our diet. That means you have to take them in. The essential ones are the ones the essential ones are the ones that are not present in our body. They are not present. Sorry for that error. The essential ones are the ones that are not present in our body. Hence, you have to take them in in our diet. The essential ones are the ones that are not present. So they are essential for your growth. So you have to take them in. Why the non-essential are the ones that are present in our diet, and hence you don't need to take them in your body. I mean, they are already present in your body. You don't need to take them in our diet. The non-essential are already present. Like something is non-essential, you don't need it because it's already in your body. Are you getting it now? So non-essential are present. The essential are not present. Hence, you have to take them in our diet. Now, having understood this, some of the amino acids can also be classified based on their polar nation. Firstly, we have what we call the non-polar amino acid. Non-polar amino acid. We have the polar amino acid. Now, under the polar, we have the non-ionizable polar amino acid and the ionizable polar amino acid. Now, when you talk about the polarity, what we mean is the nature of the side chain. Are you getting it? The nature of what? The side chain. Now, one thing that you need to know is all amino acids have from here to here to be the same. The only difference between them is the nature of our side chain. Is that okay? Let me take it again. All amino acids have from this place I put here to be the same, but the only difference is this arrow group here, which is the side chain. Now, most amino acids are optically active. Now, when we talk about optical activity, that means they have chiral carbon center, apart from few of them. Now, I will not go into classification of amino acid for you into polar and non-polar and give you structures. No. We are not going to do that. Are you getting it now? Now, the most important thing we are going to look at, let's look at some examples of amino acid. Most times, we are going to put them in their structural form. Right? In their structural form, or in their zeta ion form. So for instance, let us say you have, let me use this example. Now, recall that this group here can be anywhere. NH3, then you have C. Let's say you have hydrogen here, have COO minus. We said all amino acids assume this form, right? Where arrow is the side chain. Now, this side chain can be anything. It can be hydrogen, can be CH3, it can be anything. So let us look at the simplest, or let's say few amino acids that we are going to look at. For instance, let's say you have something like this COO minus, and then you have hydrogen, and then you have H3N positive, and then you have hydrogen. Now, this is an amino acid, and this amino acid is called glycine. Glycine is the simplest amino acid. Glycine is the what? The simplest amino acid. Are you getting it now? Now, since glycine is the simplest amino acid, look at glycine very well. Is glycine optically active? Now, we said for something to be optically active, it must have a chiral carbon center. For anything to be optically active, it must have a chiral carbon. Now, if you look at this structure very well, does it have a chiral carbon? No. Now, we talked about chiral carbon. Here we said that a chiral carbon is a carbon center that has 4 different groups about it. But if you look at this very well, this hydrogen here and this hydrogen are the same. So this one have 2 the same group. Since it has 2 the same group, it means it only have 3 different groups here and not 4. So we say this is not a chiral carbon. We looked at this when we are talking about, what is it called? Isomerism. So I don't want to dwell too much on this. Right? Now, we have other amino acids. For instance, alanine. Alanine is H3N positive, C, H, and then you have COO minus. And then, of course, you have CH3. Now, like I told you, there are about 20 amino acids. I'm not going to talk about all of them. Now, if you look at alanine, the only difference between alanine and glycine is the group here. Are you getting it now? The only difference is the what? The group here. All right. One more. All right. The only difference is the group here. If you look at this group here, you agree with me that this group here is CH3. Why the one here is what? Hydrogen. Right? So that's why we said the only distinguishing factor, the differences between each amino acid is their side chain. Now, this is a very good question that you may be asked in theory. In objective, all amino acids differ from each other by their what? The correct answer is what? Side chain. All amino acids different from each other by their what? Their side chain. Is that okay? All amino acids different from each other by their side chain. Do you understand that concept? Now, let us look at another amino acid. You have leucine, you have isoleucine. There are different amino acids that I can't even start talking about them now. Are you getting it now? So, let me give you another structure of another amino acid. This is cysteine. This is H, and then you have C, COO minus. And then, of course, you have H3N positive, and then you have CH2SH. Now, if I were you, I would just go and browse the first 20 amino acid or the 20 amino acids so that you get their structure. Now, you discover that everything here is the same, but this place here is what distinguishes them. This is what is called the side chain. Now, this form that we are writing it is what is called the zwitter ion form of the amino acid. Now, under normal circumstance, you may see this amino acid written as H, and then you have COOH, and then you have H2N, and then you have CH2 and SH. Now, there's no difference between this amino acid and this. They are the same thing. But the only difference is that this one is in what we call the zwitter ion form. I saw this in Juppé past question. That is the reason why I am actually talking about this often. This form is what we call the zwitter ion form of the amino acid. So if they say express a particular amino acid in the zwitter ion form, you write it in the form where you have H3N plus, then you have COO minus. Why in the normal form, in the unionized form, you have it as what? H2N and COOH. Is that okay? Now, we also have amino acids that have aromatic ring. For instance, phenylalanine. Phenylalanine is like aniline that has phenyl attached to it. That is why they call it phenylalanine. So what I'm going to do now is, firstly, take up the structure of aniline, duplicate it. Let me bring it down. All right. Then from here, I can get phenylalanine by removing this, and then we can erase this CH3, put CH2 here, and then draw phenyl ring. Add phenyl ring to it. Now, this structure is what we call the phenylalanine. It's an aromatic amino acid, or let's say it's an aromatic. It's an what is it called? Is an amino acid also. So you see that all the amino acid that we drew, this amino acid here is what we call cysteine, or sulfur containing amino acid. This is what we call phenylalanine, also in this zwitter ionic form. So you see that all the amino acids we've been discussing since, they have something in common. What is that? You see that all of them, they have the up in common, but the only difference is their side chain. This group here is what we call the arrow group or the side chain. So here is the arrow group here. This H here is the arrow group here. This CH3 here is the arrow group here. And of course, this one is the arrow group. And of course, this group here is the arrow group where you come to this one. So that is the amino acid. And that is one thing that you should take note of. Amino acid, they have the amino group and carboxylic acid combined together to give us the carboxylic group. Can be expressed as zwitter ion, which is dipolar ion. The zwitter ion is also called a dipolar ion. Are you getting it now? It can be expressed in the dipolar ionic form. All right. Now let's just talk about the peptide. Now, what is a peptide? Peptides are amines, the amine functional group that are formed by the interaction of two amino acids. So when you form this kind of linkage, it may be substituted. You may have it in the form of just, let's say, a bond, double bond O, and then you have NH. This is what we call the amine linkage. This is what we call the what? The amine linkage. And how is it formed? Now, we said that carbohydrates are linked together, or monosaccharides are linked together by glycosidic bond. They are linked together by what kind of bond? Glycosidic bond. Now, when it comes to amines, they are linked together by what we call the amines linkage via a condensation reaction. They are formed by the interaction between amino groups and carboxyl group of amino acid. The bond joining group, the the group together, the bond joining alpha amino group of one amino acid and the alpha carboxyl group of another amino acid is known as the peptide bond. So 2 amino acids react to form a dipeptide, 3 amino acids to form a tripeptide, and so on and so forth. So for instance, if I give you a particular amino acid, let us say I give you 2, let's say, let me just use a hypothetical amino acid like R. Let's say you have C. Hypothetical, it means no amino acid. That means you can use any of them at the end of the day. Okay, let's say here is NH2, the amine group. Here is COOH, and then you have R here. This is one amino acid, and then you have H2N combined to hydrogen, combined to COOH, and then you have R. This is another amino acid. Now, when these amino acids, when they react together, what do you expect to form at the end of the day? You are going to form the product. Now, what do you think will be the product of the reaction? The product of the reaction would be, now, it's a condensation reaction whereby water molecule is given off. Like we've talked about condensation reaction over and over again. Now, when you talk about most of the reactions of organic compounds, they are usually condensation in nature. So if we take, let's say, OH from here, right? And then take one hydrogen from here, right? We take OH from here and then take one hydrogen from here. What do you think we are going to form? We are going to form a product, and the product of this reaction would be something like this quickly. Here will give us H2N, C, H. Here is R. Here is CO. Are you getting it now? Here is what? NH. Because if you take one hydrogen from here, you know there are 2 hydrogens here. If you take one, it means you are left with one, with the nitrogen. So you have CO here, which is this CO. Then you have N and this H, which is NH. And then you have C, H, and then COOH, and then you have R. Is that understood? So this is the product of this reaction. Now, this linkage here is what we term as the peptide linkage. This linkage here, that means this group that is formed here, is what we term as what? The peptide linkage. Is that okay? Peptide. So all amino acids are joined to one another by peptide linkage. Now, this peptide, this amino acid, there are 2 amino acids that are linked here. So you call this a dipeptide. This whole compound here, you call it what? A dipeptide. Is that okay? How about if it were 3 amino acids, you call it a tripeptide, and so on and so forth. So for instance, if you combine, let's say, alanine, let's say alanine and valine, or let's say alanine and valine. Alanine is, let's say alanine and glycine. Glycine is H, just trying to combine 2 amino acid. Glycine is H2N. You have C, H, COOH. And then you have glycine. You have hydrogen here plus H2N. You have C, H, COOH, and then this is alanine. So when you have alanine and this, and you combine them together. Now, what did I say? I told you that this H2N means H and H. So when these 2 things combine, what are we expecting? If you take no. Now, if you take this group from here, OH from here, and one hydrogen from here, to form H2O, which is water molecule. So the product of this reaction we are expecting supposed to be, we are expecting something like this. So the product will be H2N, and then you have this, and then you have this, and then you have CO, and then you have NH. Recall that there's one H here. One H has been taken by this to form what? H2O. And then of course you have CH, COOH, and CH3. Plus what? The water molecule that was given off. And we said you call this a dipeptide. Why? Because it has two peptides, two amino acids. You could also have tripeptide. You could have what we call polypeptide, which are the proteins. Is that okay? So that is that for the amino acid. Now, if a number of amino acids are linked by a peptide bond, a polypeptide is formed. A polypeptide chain has an amino acid and a carboxy end. Now, what we mean is, let's assume you have this kind of a thing. Then you have so many amino acids linking together, or let's say these are amino acids. Let's just give, okay, duplicate, and then if I should take this one here, let's assume this is the group that you form. These are the amino acid, and then you have something like, let's say CO. This group here is hydrogen. Then you have hydrogen. Then you have another CO, NH. This group here is carbon, hydrogen. Let's say particular amino acid is here. This group will continue like this. This whole thing will continue. And then you have hydrogen. Let's another amino acid. And it continues like that until you have COOH. Now you discover that at the end of the day, these amino acids, this polypeptide, this is a polypeptide. Let's assume this is a polypeptide. If this chain continues, so you can call this a polypeptide. You can call it a what? A polypeptide. Now you can see that this polypeptide, what does it have at the terminal? The terminal, what it has at the terminal is an amine group at the left hand side. Then at the right hand side, what do you have here? A carboxy group. So you can see that in a polypeptide chain, it starts with an amine group and then terminates with a carboxy group. Because we told you that this group can continue like that. Is that okay? So it has an amino end and a carboxy end. That means one side of it, this is like the end to this side is amino. And then you also have the carboxylic group or carboxy end here. Are you getting it? So you should take note of that. Like I told you that the value of R is different depending on the kind of amino acid that is present. When you get to biochemistry, I think you do that in details. Is that okay? So if they give you two amino acids and they say you should join the two amino acids together to form a peptide bond, I believe you should be able to form it now, right? This process is what we call condensation reaction. I've talked to you about condensation, whereby water molecule is removed, called condensation. Are you getting? And this linkage is what we call a peptide linkage. All right? So that is that. Now let us talk about proteins. Now proteins are made up of polypeptide chain like this. This whole thing I told you is polypeptide. So most proteins are made up of polypeptide chain. Some proteins like myoglobin consist of only one polypeptide chain. Other consist of more than one polypeptide chain. Are you getting it now? For example, hemoglobin. Hemoglobin is the color imparting pigment of the blood. It also helps to transport oxygen in our body. Now, all these things, they are things that you should be doing in biology. That is a correlation between biology and chemistry and physiology, anatomy, and medicine generally. So when you understand this thing, now hemoglobin is having 4 polypeptide chain. What we mean by polypeptide chain is, now let us assume that this chain you have here is something like this. Let's assume this chain you have here is something like this. What we mean is, let's assume this amino acid is just one circle like this, like the way we have for monosaccharide when I drew one circle. Then this other amino acid is another circle. This other amino acid is another circle. This other one is another circle, another circle, another circle. It means you have so many amino acid here. This amino acid linked together to form what? Protein. Now some proteins can be just one what? Chain, like this one. Examples of such protein is myoglobin. Myoglobin is just one chain. Are you getting it now? Now, why some, for instance, insulin has 2 chains that are joined together by disulfide bond. Now, what do we mean by that? Let me erase Okay, let's see how many can go away. So let's assume you have something like this. Delete. All right. Delete. Now, this is one polypeptide chain, like I told you. Okay, let me just take it off. Example is myoglobin. It has one polypeptide chain. Now, you could also have compound that have two polypeptide chain with equal amount of polypeptides, or with equal amount of amino acid, or more than. So there's possibility that one chain may be longer than the other. For instance, let's say this chain here, they are usually joined by a kind of, for instance, insulin. Insulin has two chains joined together by what we call a disulfide bond. Disulfide is two sulfur bond. So when you have sulfur here and another sulfur here, you call it a disulfide bond. Are you getting it now? Insulin is a protein. And insulin is an hormone that regulates what? Blood glucose level. So you see protein functions in various ways. They are hormones, right? They are receptors. They are component of membranes. They have different functions in our body. Receptors, some of all these things we are taking, drugs, antibiotics we are taking, they are made up of what? Are proteins. Are you getting it now? Enzymes are protein. Say, what are enzymes? Enzymes are organic catalysts, proteinous in nature, right? So many things that we have to talk. We talk about the dry body weight contain mostly proteins. Are you getting it now? Most tissues are proteinous in nature. Is that okay? The cell contains about 65% of protein. So if 65% of our body, of our cell, you know, the cell is the basic building block of the full body. So if the entire cell, okay, the cell membrane, not even the entire cell, the cell membrane contains about 55% protein. Most component of our body, 78% is almost about what? Proteins. So that is that. Now, insulin has two chains joined together by what we call a disulfide bond. Disulfide means two sulfur linkage. Now, another thing that you also need to know, like I told you, that these two chains can be of the same number of amino acid, or they have various different number of amino acid. So this one of these chains is called a polypeptide. Are you getting it? This is also a polypeptide. So when they combine together, you can actually have different names that you call them. All right. So, for instance, hemoglobin that helps to transport oxygen in the body and give blood this color has 4 chains. 2, one kind of 2, and 2 of another. That means 2 the same kind and the other 2 different chain. Now, the amino acid sequence in different kinds of polypeptide chain of a protein is specified by the gene. Are you getting it now? It's specified by the word, the gene. So, and a difference in the genetic makeup of this polypeptide chain. The polypeptide chain in most cases is also called the primary structure of the amino acid. The order at which these amino acids are arranged is determined by the gene. And any shift in the position of any of these amino acid, or a replacement of one amino acid by another, is what we call genetic mutation. That means what? When you say mutation, means aberration, genetic change. Are you getting it now? So the sequence of amino acid you have here is specified by the gene in the body. Your gene is like your blueprint. Are you getting it now? That is what specifies the nature of these amino acids. Are you getting it now? So we can say the sequence of amino acids. For instance, if this is alanine, valine, glutamine, asparagine, and different amino acids, it is specified by the gene. And any change in this will cause serious illness and disease. That is what causes sickle cell anemia. Just a change in one of the amino acids from glutamine to one like that. Think glutamine and valine or leucine. That causes sickle cell anemia, which is SS. They tell you it's the genetic disease. Are you getting it now? So that is that.